Welcome to On the Shoulders of Giants, Interpreting Gene Expression Results in the Context of Published Biological Findings. I'm Tamlin Oliver, Managing Editor of Genetic Engineering and Biotechnology News, and I'll be the moderator for this webinar. The proliferation and complexity of gene expression data resulting from high-throughput real-time PCR, microarray, and RNA-Seq technologies offers an abundance of research opportunities, but presents several challenges to getting a rapid, accurate biological understanding of data. To address these challenges, Ingenuity Systems has developed Ingenuity iReport for real-time PCR, microarray, and RNA-Seq analysis. In this webinar, industry thought leaders will demonstrate this integrated statistical and biological analysis strategy using two different gene expression data types. The three speakers today, Michael Kubista, Milan Radovic, and Gordon Janaway, will share their experiences and insights in just a minute. Before we get started, though, I want to encourage you to submit questions for our panelists. We'll answer as many questions as we can after the final presentation. Simply type your question into the Ask a Question box and hit Submit. Michael Kubista, professor at the Tata Center, will discuss his real-time PCR study of single-cell astrocytes. In his presentation, you'll see how multivariate analysis was combined with Ingenuity iReport to characterize subtypes and changes induced under conditions such as aging and healing after brain damage. Michael, we're ready when you are.
Thank you, Tamlin. Good morning. My name is Michael Kubista, and today I will present our latest developments in qPCR expression profiling workflow. Next slide. I am from the Tata Biocenter in Gothenburg, Sweden. Tata Biocenter was founded in 2001 around competence in real-time PCR by researchers that pioneered the field. Today, Tata Biocenter is Europe's leading provider of qPCR services. Tata also organizes hands-on training, and we are involved in the standardization of the pre-analytical processes in molecular diagnostics for Europe. Tata pioneered many areas in the field of qPCR, including single-cell profiling, high-throughput and multi-way qPCR profiling, qPCR tomography, and aspects of immuno-qPCR and digital PCR. In our work, we are supported by leading instrument and reagent companies in the qPCR field. Next slide. One of the complexities in molecular diagnostics is the heterogeneity of tissue. This is illustrated in this slide, showing an islet of Langerhans. The islet is stained for delta cells. Next slide. This shows the same islet stained also for the alpha cells. Next slide. And here is the islet stained also for the beta cells. There are other less abundant cell types as well. Of importance here is that the islet is a mosaic of the different cell types. Each cell type has a distinct biological function and response to certain stimuli. Beta cells, for example, respond to glucose and produce insulin. If you are interested in glucose stimulation of insulin and wish to study this on the molecular level, we are facing the problem that analyzing an islet or even a piece of the islet, we get a mixture of cells of which only a fraction of beta cells that respond to the stimuli. All the other cells only contribute with background and confound the measured responses. To approach the complexity of tissue heterogeneity, we developed single-cell qPCR profiling. Next slide. Disintegrating an islet and sorting the cells, we measured the number of beta-actin transcripts in some 100 individual beta cells. The data are presented in a histogram showing the distribution of beta-actin transcripts among the beta cells. Some 40 cells had less than 100 beta-actin transcripts. 25 cells had 100 to 200 transcripts. 15 cells had 200 to 300 transcripts, and so forth. Only very few cells had high levels of beta-actin transcripts. This highly skewed distribution of the number of transcripts among cells was found also in cultured beta cells. Next slide. Similar results were observed for other transcripts and later also for other types of cells. Today, we know that at any given time, even in a seemingly homogeneous cell population, most cells have few transcripts only, and high transcript levels are found in a minority of the cells. Next slide. Showing the same data, but in logarithmic scale, we find that the spread of transcripts among the cells is consistent with a normal distribution. Hence, the distribution of transcripts among like cells is log normal. Next slide. Log normal distribution of transcripts has several consequences. For example, the number of transcripts in the typical cell is given by the geometric average of the number of transcripts per cell, rather than by the regular or arithmetic average, which is obtained as the total number of transcripts measured in a traditional sample divided by the total number of cells. Next slide. As pointed out by Nature Genetics in their perspective of our paper, the difference in the two means can be confusing as well as essential. Next slide. Already in our first study, we measured the expression of multiple genes per cell and could compare the abundance of the different transcripts in the individual cells. The results are summarized here in a table as correlation coefficients. A correlation coefficient of 1 indicates that the two transcripts are present at equal levels in each cell. A correlation coefficient around 0 indicates that the two transcripts are produced independently of each other in the cell. And a negative correlation would indicate that when one transcript is present in a cell, the other transcript is not. Next slide. Inspecting the correlation coefficients, we find only a single large number. This is the correlation coefficient of insulin 1 and insulin 2, indicating that these two transcripts are present at equal levels in each of the mouse beta cells. This makes perfect sense considering their biological function, since the two transcripts are needed in equal amounts to produce native insulin. The correlation coefficients between all other pairs of transcripts are not significantly different from 0, evidencing that they are present independently of each other in the beta cells. Next slide. Hence, different beta cells contain different transcripts. Work from other laboratories has shown this difference is dynamic. Genes are transcribed in bursts, and the particular transcripts present in a cell vary over time. Next slide. We have developed a high-throughput workflow for single-cell qPCR expression profiling. Cells from culture, blood, or disintegrated tissue are collected with a fax into a special lysis reagent that is compatible with downstream RT-qPCR. The RNA is reverse transcribed and the cDNA is preamplified by multiplex PCR for a limited number of cycles. The amplified material is aliquoted and quantified by parallel singleplex qPCR. The cells are then classified based on the measured expression profiles with multivariate methods using the GENNews software. This workflow has proven exceedingly powerful. Next slide. In this talk, I will take the workflow one step further, correlating also the measured expression profiles to biological properties and functions. Next slide. The study I describe is of astrocytes in mice using GFAP-enhanced green fluorescent protein strain, which allows us to identify and collect astrocytes from the disintegrated brains using fax. We study astrocyte function in ischemia during aging and also the role of astrocytes in repair. Next slide. This slide shows distribution of various transcripts among individual astrocytes. At first inspection, we recognize the log-normal distributions described before. Next slide. However, closer inspection reveals exceptions. Vimentin, for example, shows distinct bimodal distribution, evidencing heterogeneous population with subtypes characterized by either low or high vimentin expression. Next slide. The distinction of cell subtypes can be improved using multivariate methods. Here we classify individual astrocytes collected from control and mouse brains 3, 7, and 14 days after injury. The classification is based on principal component analysis of the expression of 47 genes. There is overall large spread of the astrocytes in the plot, which reflects the natural heterogeneity among like cells. But the blue controls are located predominantly in the top left corner. Astrocytes collected 3 days after injury are shown in yellow and are located somewhat lower down and a little bit to the right of the controls. Astrocytes collected 7 days after injury, shown in green, or even further down and to the right. Astrocytes collected at 14 days after injury are shown in red. All but 2 of these astrocytes are found clearly separated in the top right corner of the scores plot, evidencing they have distinct expression profile, presumably due to activation of genes induced by the injury required for the healing process. Next slide. The genes important for the activation can be identified by classical comparison of genes expression in the astrocytes 14 days after injury and those from the control. Differential expression is shown in the bar plot. The p-values indicating significance are found in the table, and the 2 parameters are combined in the volcano plot for easy recognition of important genes. These are located towards the upper corners. Next slide. This straightforward analysis of one gene at a time has, however, major drawbacks. Because of the large number of parallel comparisons performed, there is ambiguity with high false positive rate and low power. Genes that are not relevant may incidentally have low p-values, and genes that are truly differentially expressed may remain unnoticed. Next slide. Better is to take advantage of genes' correlated expressions, identifying those that are collectively activated in the astrocytes. These genes are identified in the principal components loading plot. Next slide. These genes are expected to be important because of their collective changes in expression at 14 days after injury rather than individual changes. Next slide. The information content in a PCA presentation can be enhanced by showing the data in a 3D graph. Frequently, the 3D graph reveals additional features of the analyzed cells. Next slide. Rotating the 3D graph into position, we find that the activated astrocytes divide into 2 clusters, evidencing there are 2 kinds, characterized by different expression profiles. Next slide. Calculating the correlations between the genes' expressions in individual astrocytes, we identify groups of genes that are active at the same time in the same cells. These genes are likely to produce transcripts coding for products required in common pathways. Note, these pathways are identified based on correlated expression in single cells rather than on correlated expression in bulk, which may be indirect due to independent pathways being activated by a stimulus. Next slide. The calculated correlations can be decomposed into direct and indirect correlations to identify master genes. Next slide. To relate the observed expression patterns and correlations among genes expressions in individual cells to function, and also to identify complementary genes that may be relevant for the phenomenon we study, we submit the data to Ingenuity, generating an iReport. Based on the genes we have submitted, relevant pathways, processes, and diseases are identified. Next slide. The results are summarized in the wheel, where we can choose to indicate, for example, genes' molecular functions, processes they are involved in, and relevant expression pathways. Next slide. Focusing down on diseases, we find, not surprisingly, that many of the genes we identified as important for astrocyte activation have previously been related to schizophrenia, Alzheimer's, and seizures. Next slide. To identify additional genes of relevance, we perform pathway analysis. The iReport presents the pathways our genes are involved in. For example, 5 of our genes are involved in glutamate receptor signaling. Next slide. Inspecting the glutamate receptor signaling pathway, we identify other genes that may be of relevance. Next slide. We also find important publications in the iReport. Next slide. This information leads us to select additional genes to include in our single-cell profiling study to elucidate the mechanism of astrocyte activation. Next slide. In conclusion, I have presented workflow for high-throughput single-cell expression profiling by quantitative real-time PCR correlating single-cell expression profiles to biological function. The advantages are that complexity caused by the heterogeneity of traditional samples is avoided, and high power to identify relevant genes is obtained by correlation. The cell's profiles are related to function by mining a powerful database. Next slide. The experimental work has been done by my co-workers Vendela Rushniakova at the Institute of Biotechnology and Mirka Angerova at the Institute of Experimental Medicine of the Czech Academy of Sciences in Prague, and Anders Ståhlberg at the Center of Cancer Research, and Lars Pekny at the Institute of Psychology and Neurosciences at University of Gothenburg in Sweden. Next slide. Finally, I also thank MultiD for providing me with early access to their latest version of GENNews software for expression profiling, and to Ingenuity for providing early access to the iReport for qPCR data mining. Thank you for your attention. I'll be happy to answer questions.
Thanks, Michael. Before we move on to our next speaker, I want to remind you that you can submit questions at any time by typing the question into the Ask a Question box on the lower left of your screen and hitting Submit. Our second speaker is Milan Radovic, Assistant Research Professor of Surgery at Indiana University School of Medicine. In his research, next-generation RNA-Seq data was combined with iReport to compare the transcriptomes of triple-negative breast cancer and normal breast samples. His group was able to show how to identify differentially regulated pathways and candidate drug targets for future investigation. Milan, you can begin now.
Thank you, Tamlin. Well, it's a pleasure today to present to you my seminar on next-generation sequencing in triple-negative breast cancer and normal breast tissues. This work here is going to comprise a little bit about our analysis of using RNA-Seq data in triple-negative breast cancer versus normal, and then how we integrated network analysis to understand some novel findings. Next slide. I'd like to first start off a little bit with the definition of what triple-negative breast cancer is. This flow diagram here starts at the top with a picture of one of our patients here at the IU Cancer Center. When we have patients who come into the cancer center, we'll get a biopsy of their primary tumor. And as with all breast cancers, this primary tumor is going to have a series of tests done, immunohistochemistry-based tests, to determine positivity for a set of different proteins. The first set of proteins that we will look at is the estrogen receptor and progesterone receptor. This comprises about 75% of all breast cancers. And if a breast cancer is positive for the ER or PR protein, in addition to standard chemotherapy, patients will receive targeted agents that target these proteins, which include tamoxifen, fulvestrant, or the aromatase inhibitors. A second protein that will be tested for on a tumor biopsy will be the HER2 protein, or the human epidermal growth factor receptor 2. This makes roughly around 10 to 15% of all breast cancers, and if it is overexpressed in the sample, in addition to chemotherapy, we'll receive drugs that target the HER2 protein, specifically the antibody Herceptin or the small molecule lapatinib. What we then have is this other set of samples, or this which also make about 10 to 15% of the breast cancer population, which are negative for the ER, PR, and HER2, and we refer to these as the triple-negative population. And in addition to chemotherapy, there is no targeted therapies for this patient population. Next slide. What this slide shows is a Kaplan-Meier curve of all other breast cancers versus triple-negative breast cancers. As you can see that having this paucity of targeted therapy, patients with triple-negative have poorer prognosis compared to all other breast cancers in terms of overall survival. Next slide. As a little review, triple-negative breast cancer, as I mentioned, they make up 10 to 15% of all breast cancers with a disproportionately higher mortality. They tend to affect premenopausal women and women of African descent. As many of you know about the BRCA1, which genetic mutation predisposes to women with breast cancers, and if a BRCA1 carrier does develop breast cancers, they tend to be triple-negative. Also, as mentioned, there is a paucity of effective targeted therapies. As such, these patients don't respond to estrogen blockade or anti-HER2 therapies. And I think the bigger question is, as scientists, where can we look to better define this cancer, triple-negative breast cancer, and also define new therapeutic targets? Next slide. So in our pilot study here I present the flow diagram where we took 10 triple-negative breast cancers from premenopausal women and also 10 normal breast tissues from our Susan G. Cohen for the Cure Tissue Bank here at the IU Cancer Center. We depleted the samples for ribosomal RNA and then sequenced the RNA-Seq chemistry on the Applied Biosystems solid system. The reads that were produced by the solid system were then mapped to the human genome using the ABI Bioscope 1.2 software, and then outputs from the mapping were analyzed with both in-house software from our Center for Computational Biology and Bioinformatics, as well as third-party software from Applied Biosystems, Biotech, and Ingenuity Systems. Next slide. One important part of our study is the use of our normal controls. In our case, our normal controls came from healthy women in which core biopsies were taken, and then these biopsies were then frozen. Because the origin of breast cancer is the milk ducts, or the epithelial component, and this component makes up a minority of the cells within the breast, we undertook laser capture microdissection in order to just isolate RNA from the epithelial component of the normal breast. And here, an example, here on the left, we see what the tissue looks before microdissection. The cells in brown are the stroma, the fibroblasts, and here in the middle are the, as you see with these little luminal, this tubular type cells, in blue are the milk ducts. Using laser capture microdissection, we then cut around the milk ducts, extract them from the cap, and then extract the RNA. Next slide. So some of our first analysis here, presented here, which is a principal components analysis of the gene expression values derived from the RNA-Seq. For those who are not familiar with principal components analysis, each little sphere here represents one of the samples, one of the 10 triple negatives in blue, or one of the 10 normal samples in red. And essentially what this does is plot how samples are alike or dislike based on gene expression values within a three-dimensional space. And what we can see here is a vast separation of the gene expression profile between triple-negative breast cancer and normal, and what this translates into is roughly 5,600 genes are significantly different between these two types. Next slide. Well, one question that many would have would say is, well, why do you need RNA-Seq? We have done microarrays in breast cancers for over a decade. here the very famous work done at UNC in which breast cancers using microarrays were separated into 5 intrinsic subtypes, particularly known as the luminal A and luminal B, which are the ER-positive subtypes, HER2, which make up the HER2 subtype, the basal-like subtype, which is very prominently triple-negative, and then the normal breast-like. And then the controls in these experiments were the use of reduction mammoplasties. Though important part of these previous studies was the normal control, the use of reduction mammoplasties we know is comprised mostly of adipose tissues. When one analyzes or looks at these papers, we see high expression of adipose genes, such that when we do comparisons between breast cancers, what we have primarily in these previous studies are tumor versus tumor comparisons, such that one subtype of breast cancer is then compared to another subtype versus to normal. One of the problems with doing tumor versus tumor comparisons is that it increases the likelihood of what we call passenger findings. Because there's a difference between tumors, one breast cancer to another, doesn't necessarily mean that that difference is important. It could be just a passenger finding. Whereas when we do a comparison of triple neg to normal, for example, by definitions, all differences have the potential, and I do importantly stress the potential to be important. Next slide. Well, how does this translate when we look at the clinic? This table here is a table of targeted agents tested in clinical trials of triple-negative breast cancer. On the left, you see the targeted agents listed. The next column shows the protein targets of these agents, and then what we saw in RNA sequencing, as well with the fold change, and then the clinical trial outcomes. First target is EGFR, which has been tested with the drugs cetuximab and gefitinib. And our previous data has shown that the EGFR was to be overexpressed, triple neg versus other breast cancers. When we compared triple neg to normal, we found it not to be overexpressed, p-value insignificant, and all these clinical trials, 4 of them now, were negative. The next set of drugs, imatinib and dasatinib, which target the c-KIT receptor, also previously reported to be overexpressed in triple-negative. We found it to be not overexpressed, actually significantly downregulated, at a very significant p-value, and 2 clinical trials testing these drugs have been negative. And the last one we look at is nipirib, which is a PARP inhibitor. We found PARP to be overexpressed in triple-negative versus normal, a 4.3-fold of highly significant p-value, and we saw a positive phase 2, though in the phase 3, the primary important point was negative with some activity in the refractory setting. That being said, several PARP agents are now in clinical trials showing some promising results. We will see results of phase 3s here in the near future. Next slide. Well, one of the major questions then would be, are the results we're seeing real? For example, are the results we're seeing with the EGFR, KIT, and PARP findings I just presented an artifact of sample size or possibly technology? So we then underwent a separate validation cohort of 26 additional frozen triple negatives and a separate set of 10 microdissected normal tissues, evaluated RNA expression by the gold standard, ABI-TACMan assays, and then also followed up at the protein level by immunohistochemistry. Next slide. And what I'm showing here is the qPCR results of the EGFR, KIT, and PARP1, the normals, the gene expression of the normals in red, the gene expression of triple neg in green, and as we work from left to right, the EGFR not differentially expressed, significant p-value. With KIT, we see triple negative significantly downregulated with a highly significant p-value, as we saw in the RNA-Seq, and then also with the PARP, we see the triple negative overexpressed compared to the normals with a highly significant p-value, again, validating the results that we saw from the RNA-Seq. Next slide. We then also went to do immunohistochemistry. As some will be familiar, EGFR and KIT are standard immunohistochemistries done in hospital laboratories around the world, as well here at IU. So we decided to send these same samples to the same hospital laboratory where patients would have their EGFR and KIT immunohistochemistry done. And here we see, again with EGFR, with normals in blue and triple neg in red, we see no differential expression based on H-score of EGFR and significant p-value. And then with KIT, we see a significant downregulation, just like we saw in the qPCR and the RNA-Seq significant p-value. Next slide. This is a little bit of a busy slide, but this is the immunohistochemistry that we've seen for the EGFR and KIT. On the left side is the EGFR. We see expression just very widely in the normals. Also in the triple negs, we see some super high expression there in the top panel, and then we see no expression in the next one, as well as very little expression in the other panels. When we look at the right side of the slide where we see KIT, we see in the normals, we see a very high expression of KIT, especially there in the luminal ducts and across all 4 panels in this case, and then in the triple neg, we see a varying expression, but in general, actually pretty absent expression. Next slide. So this actually begs a question. EGFR and KIT belong to a class of proteins known as receptor tyrosine kinases. As many would know, receptor tyrosine kinases are key proteins on the cell surface that activates a majority of important signaling cascades in cancer. When we had done some network analysis, we saw several proteins downstream of RTKs that seem to be overexpressed, but which leads to the question, though, which RTK is important here in triple negative if it's not EGFR and KIT? Next slide. So here, what we did was we found about roughly 50 or so receptor tyrosine kinases in the genome. About 32 of these were expressed in our data set, and we then plotted our expression data on a volcano plot, where on the x-axis we have the fold change, going from a negative fold change to a positive fold change, and then on the y-axis we have a p-value of the statistical significance of the differential expression between triple negative and normal, such that on a volcano plot, the genes at the left tip of the V represent the most downregulated genes, and the ones on the right tip of the V, the most significantly upregulated genes. And what we noticed here is a set of receptor tyrosine kinases that seem to be highly overexpressed in triple negative versus normal, whereas KIT and EGFR were either highly underexpressed or not differentially expressed respectively. One gene in particular that was of real interest was our top hit here, as signified by the blue arrow, was PTK7, or protein tyrosine kinase 7. Next slide. This is a very, very interesting gene. It was first discovered as a protein expressed in colon carcinoma, but not normal colon tissue. It is a key regulator of epithelial development. This is very, very interesting, as we know that cancers, and specifically triple-negative breast cancer, has a very stem-like phenotype, and to see a gene involved in development was very exciting for our group. We also know that PTK7 mutations cause defects in mouse embryos, particularly in the heart, lung, and ear development. To further support our hypothesis here, a paper published in CCR Clinical Cancer Research in 2009, in which PTK7 was actually a gene that was knocked down in ER-positive cell lines here on the left, and then ER-negative cell lines on the right. If you look at the details of these graphs, what was shown was that by knockdown of PTK7, we saw a significant inhibition of cell growth and proliferation, specifically in the ER-negative cell lines, but not in the ER-positive. And this further gives evidence to the credence of PTK7 being an important player here in ER-negative and specifically triple-negative breast cancer. Next slide. So this then led us to say, well, if we see this sort of differential here in receptor tyrosine kinases, how about all kinases? So here, using Ingenuity's iReport, we imported our data into this server and decided to look at kinases that are overexpressed in our data set, but are highly connected based on their interactions of the Ingenuity database. Here, I'm presenting to you what is referred to as the wheel here in the iReport, and each one of these circles represents one of the 276 kinases that were differentially expressed in our data set. Those in yellow are overexpressed, and those in blue are underexpressed. The circles then are sized by the number of molecular interactions that are known in the Ingenuity database. And our top hits here were I kappa B kappa epsilon, the pro-survival protein AKT, the known oncogene SARC, and also cyclin-dependent kinase 1A, or also known as P21. Next slide. Building on that data, seeing that AKT, a well-known survival protein cancer, was overexpressed, we decided to look at the AKT pathway, again here using Ingenuity pathway analysis, and we see here AKT at the center to be overexpressed, and as well as a cadre of many genes both upstream and downstream of AKT also being overexpressed. What was very interesting, you may remember from the last slide, was also I kappa B kappa epsilon being a highly connected overexpressed kinase, and to our surprise, AKT is also activates IKB kappa epsilon via COT, and is represented also by my red arrow in our laboratory now is looking at several knockdowns to see how this will affect our cell phenotype. Even further importance, though, is that the AKT inhibitors have now just recently entered phase 1 and 2 clinical trials, so even in breast cancer, and it will be interesting to see how this particular drug target will turn out in the clinic. Next slide. So, as I mentioned during the introduction, that BRCA1 is a very important protein involved in triple-negative breast cancers. As I mentioned, the majority of BRCA1, if they are mutation carriers, are going to be predisposed to triple-negative breast cancer and also ovarian cancer. BRCA1 is an important protein involved in DNA repair via homologous recombination, and we know that those patients who are tumors that are mutated for BRCA1 are defective in HR repair. We also know that triple-negatives that are not mutated for BRCA1 do still exhibit defective DNA repair, and this is referred to by umbrella term referred to as BRCA-ness. Next slide. In our data set of really strong interest, our number one statistically upregulated gene in our data set was a gene called COBRA1, also known as cofactor of BRCA1. This gene was first discovered in a yeast 2 hybrid screen as a protein that physically associates with BRCA1, and as mentioned, the majority of BRCA1 breast cancer are triple negative. It is also a member of a complex known as the negative elongation factor complex, which we know inhibits RNA polymerase transcription of genes of estrogen receptor positive genes. Further data by another group also showed that siRNA knockdown and microarray analysis in T47D cells found to regulate 287 genes are also regulated by BRCA1. So there's a huge overlap of the genes that BRCA1 and COBRA1 co-regulate. What I think is probably the most interesting when we look at this data is that our number one statistically significant upregulated gene in our data set is involved in both negatively regulating estrogen receptor and BRCA1. Here we have a triple negative breast cancer, again, that doesn't express the estrogen receptor and also has known predisposition to BRCA1. This then led us to really try to interrogate more of the BRCA1 pathway. Next slide. So again, here using Ingenuity pathway analysis, we imported our data into Ingenuity and then took a look at the BRCA pathway. Here, genes in red are those that are overexpressed compared to triple negative versus normal. And we can see a variety of genes, both upstream and downstream of BRCA1, are overexpressed. Of even further interest were the genes that are circled in gold. These genes represent targets that are now actively in clinical development and hopefully soon to enter clinical trial. It would be interesting to see also the effect of these overexpressed targets in triple negative in the clinic. Next slide. Another analysis that we then performed was using a new tool available by Ingenuity called the transcription factor analysis. Here we import our differentially expressed genes into the Ingenuity software, and the software then, based on our gene expression, will determine what transcription factors are activated or inhibited based on the direction of our expression. This is actually a screenshot from that analysis, and the genes of interest are highlighted in blue. For example, our very strongly activated transcription factor was MYC. MYC is a well-known oncogene in cancer, but was even more further interested was the ones that were inhibited. The ones there in blue, TP53, retinoblastoma, RB1, and then CDKN2A, also known as P16. First, P53 was extremely interesting as the majority of triple-negative breast cancers, over 80%, are mutated for the TP53 gene. And to see that its downstream targets were inhibited was of great interest. In addition, retinoblastoma, there is some data suggest that a small portion of triple negative also have somatic mutation for retinoblastoma, but it may seem that this inactivation of retinoblastoma is not also restricted to mutation, but at the expression level, seemingly that RB1 is not acting normally. Here also then CDKN2A is a great positive control. It's well known in many cancers for P16 to be overexpressed, but not to be active in the cancer. Next slide. So then, building upon these P53 findings, we also did a network analysis using Ingenuity of P53. Here we also combined some data that we garnered from the RNA-Seq of precursor microRNAs expression. And here, sort of preliminary analysis, we did a combined precursor microRNA and P53 analysis. And what was very interesting here is the genes, the microRNAs here in red that are overexpressed, and here ones in the green are underexpressed. And if you were interacting interactively with the Ingenuity software and you were to look at these interactions, for every microRNA that's overexpressed, if P53 was acting normally, would actually be underexpressed. And those that, all the microRNAs that you see in green that are underexpressed, if P53 was normal, non-mutated, these would actually be overexpressed. So they're always acting opposite the direction of what we expect normal P53 to do. Again, further confirming the findings that P53 here in triple-negative breast cancer is inactive. So next slide. Well, this work is just obviously the conglomerate of many, many great collaborators, but all here at the Indiana University School of Medicine, also the Susan G. Komen for the Cure at the IU Summit Cancer, as well as Applied Biosystems and Cofactor Genomics. And that's the end of my presentation. Back to you, Tamlin.
Thanks, Milan. I just want to remind you one more time about the questions. If you have any questions for Dr. Radovich, please submit them now. Our third and final speaker is Gordon Janaway, Senior Product Manager for iReport at Ingenuity Systems. Gordon will talk about using iReport to rapidly and accurately identify key findings in gene expression results and evaluate them in the context of the most relevant information from the published biological literature. Gordon, we're ready when you are.
Thank you, Tamlin. My name is Gordon Janaway. I'm a Senior Product Manager at Ingenuity Systems, and I work on a product called iReport. I'm here to talk today about interpreting gene expression results in the context of published biological findings. Next slide. Today, with high-throughput qPCR, microarray, and RNA-Seq technologies, we are experiencing a dramatic increase in the quantity of gene expression data available to researchers. And this abundant data presents both challenges and opportunities to biological researchers. On the one hand, more expression data is available than ever before, but on the other hand, important biological relationships can be lost in a sea of publications, and new insights from data that you generate in your own lab can be obscured by burdensome and complex statistical analysis. So many of us are suffering from a problem of too much data and not enough biological meaning associated with that data. Next slide. First, I'd like to describe a general framework for understanding gene expression analysis. This framework applies to RNA-Seq data, microarray data, and real-time PCR data. And I think you'll be able to see how many of the assumptions that go into processing this data can lead to somewhat of an error-prone process that can have arbitrary inputs and that also can be quite time-consuming. So on the right of this slide, you see three basic steps that are involved. Generally, the raw data that is obtained during a gene expression experiment needs to be normalized for the quantity of sample that was loaded into that experiment. And in every platform, there are various thresholds that can be adjusted that significantly affect the output of the analysis. And then whatever experimental conditions were used in the design of the experiment can be compared, and fold changes can be calculated, significance can be estimated from p-values, and from those values, then key genes can be identified. And then once those genes are identified, then the researcher can begin to look into the literature to understand some of the biology that they did not already understand about those genes. Next slide. So I'm going to choose real-time PCR as an example. So during the normalization and threshold process, CT values or CQ values are adjusted according to maximum thresholds. Outliers may be removed from technical replicates before averaging CT values. And then one pain point for many real-time PCR customers is the selection of endogenous controls used to normalize their data to produce delta CT values. So often the selection of these controls has a dramatic impact on the end result of the analysis, and the scientists don't know how to normalize well or how to assess the quality of the normalization. Next slide. After normalizing the data, then the different conditions that were set up in the experiment can be compared. And for real-time PCR, this means calculating delta-delta CT values is a very common way to do this. And then from delta-delta CT values, they can be converted to fold change, and also p-values can be calculated. And then from those values, you can begin to identify interesting genes within your real-time PCR experiments. Next slide. And then after that, often researchers will search the literature for context to try to understand the results of their experiment. And at that point, they can finally get to the interesting place where all of us would like to be, which is interpreting and understanding the biology that's going on in the system that we're studying, using the literature to stand on the shoulders of the giants who have come before us and published so much on this system, to understand the biology, develop new hypotheses that are truly new, and also to share the results, whether it's in publication, a poster, a presentation, or even just within a laboratory. Next slide. So to meet these needs, Ingenuity has introduced a product called iReport. And iReport is the fastest way to understand the biological meaning of your data. We have designed iReport with 4 characteristics in mind. Fast, meaning that a straightforward upload and analysis of raw data to quickly see beyond the statistics to the biology. So this is taking data directly from the instrument that's producing the data and analyzing it to quickly get to the biology piece of the analysis. The iReport is accurate. The findings that are produced in iReport are supported by evidence in published literature or in commonly used databases. And every finding that is presented in iReport is provided with a link so that you can always access the primary information that provided that finding. iReport is interactive, so it is not a static report, but it's a web-based report that allows you to visually organize and filter your results relevant to your experiment. So instead of receiving a mountain of publication and data relevant to the genes that you've studied, we have provided a customized product that is unique to your data set. So each iReport is unique to a specific data set, and that iReport summarizes not only the statistics but also the biology that is going on in that data set. So on the right of this slide is an image of a user interface which we describe as the wheel, which allows differentially expressed genes to be displayed as colored circles in this image, and they can be grouped by molecular function, for example, or and colored by expression level. We'll be talking more about this interface in a minute. Next slide. I want to just give an overview of iReport specifically for real-time PCR. And here are some of the steps that the iReport upload process walks you through. And these steps, again, are required for real-time PCR in general. And our goal in building this product was to try to streamline this process as much as possible. So you would first upload your CT data, then define the condition and the control samples that you would like to compare. Select your endogenous controls for normalization. And then the primer probe sets, or the primer pairs that you used in your qPCR experiment, need to be mapped to genes in order to generate the biologically relevant results. So this mapping is done automatically when possible in our system. And so at this last stage, you confirm the mapping, and then you can view the iReport. Next slide. So I want to walk briefly to a couple of these user interfaces. So one of them is assigning endogenous controls. You can see here in the top left is the list of all the assays in the experiment. And then on the right are two candidate control assays. And the variation in these expression levels of these assays across the different sample values is shown in the graph below. Next slide. And as you can see in the center, you can add and remove these endogenous controls from the list and then view dynamically the expression levels across the different samples. Next slide. This is the interface for mapping assays to genes. As I mentioned, if your assay name is not recognized by iReport, then you can map these assays manually. Next slide. See on the left is the assay list from the uploaded file. In the middle is the automatic mapping of many assay types, in particular TaqMan assay IDs, and the formats that are used with TaqMan low-density array cards are automatically mapped using this setup tool. And then those genes are mapped to homologs which are present in the iReport. And if a particular assay is not mapped, then you can enter that gene here, and an auto-population field will pop up so that you can map that correctly. And finally, you generate an iReport. Next slide. So I want to give you just a brief overview of iReport, and then we'll go into a case study. So this is the first page of a multi-chapter report. We define these different tabs as chapters. Next slide. On the lower left, you can see a volcano plot. The volcano plot indicates the fold change and the significance, the p-values of the gene expression data. And also you can see a list of the genes sorted by the connectivity, so the degree to which that gene is connected to other differentially expressed genes in your data set. Next slide. On the lower right is a list of the most significant biological pathways that we have found relative to this data set, the most significant processes, and the most significant disease states or data about diseases that are relevant to these results. Next slide. As I mentioned at the top, you can see that there are several tabs, and we call these chapters, and I'll be describing these various chapters in the coming slides. Next slide. First, I'd like to introduce a case study. I think it's more relevant to speak about a specific example. This study was just published in the journal Blood, and it's a study of Notch promoting vascular maturation by inducing integrin-mediated smooth muscle cell adhesion to the endothelial basement membrane. This data set was uploaded into GEO, the Gene Expression Omnibus, and this was a real-time PCR data set, and we have analyzed this data set. Next slide. So just some brief background on the science behind this publication. Interaction with JAG1 is known to activate Notch signaling in vascular smooth muscle cells, or VSMCs, leading to arterial maturation. The exact mechanism, however, of Notch-mediated maturation is not known. So the authors knew that JAG1 would activate Notch, and that Notch led them to the formation of the recruitment of these smooth muscle cells. But the mechanism of that recruitment and the maturation was not clear. So the objective of the authors was to identify genes involved in the molecular mechanisms of JAG1-mediated Notch signaling for recruitment and retention of VSMCs in vessel maturation. And in their paper, they discussed several methods that they used to address this question, and one of the methods was real-time PCR. So they grew these VSMCs in different plates. Some were coated with recombinant JAG1, which would activate the Notch pathway. And then real-time PCR was used to measure expression differences between VSMCs that were cultured with and without JAG1. Next slide. Just a little bit more about this real-time PCR experiment. So 18 hours after the culture, the researchers isolated the RNA and produced a cDNA. The cDNA was mixed with reagents and loaded onto a Tagman low-density array card. This is the human angiogenesis panel provided by Biotechnology or Applied Biosystems. And it was run on a 7900 real-time PCR system. And then the fold change was measured for each gene calculated, and this fold change was uploaded into GEO. Next slide. So here's the summary page for the iReport that we generated from this data. And you can see right away that there are quite a few pathways, processes, and diseases that are relevant to this data set. Next slide. In particular, the top-ranked pathways include angiopoietin signaling and Notch signaling. The top-ranked processes include migration of endothelial cells and vasculogenesis. And the top-ranked diseases include vascular disease. So many of these results are perhaps obviously related to the hypotheses of the experiment. And those are the ones that I've highlighted here, the endothelial cells, the vasculogenesis, and so on. Others may not be immediately obviously relevant, but these are the results that come up through the automated processing of the data. And our goal in presenting this information is to rank these results as intelligently as we can, and then provide a transparent interface for the researcher to quickly identify the relevant processes, pathways, and diseases for their research. Next slide. In particular, for Notch signaling, we knew that that was going to be a topic of study, and it does appear in the results. If we go to the Notch signaling link, then we're taken to the Pathways chapter, and here you can see several things. Next slide. One of them is the Notch signaling pathway itself, which I will show again in a minute. And this is shown in the center of the page, and that can be exported as well. That image can be exported. As you can see in the center of that page, only one gene in that pathway was interrogated in this real-time PCR experiment. So that suggests that future experiments to study this particular pathway, this is activated by JAG1, as you can see in the pathway image. It may be useful to interrogate some of the other genes in that pathway. On the lower left is an overview of the pathway, describing generally that pathway and the genes involved in that pathway, which were assayed in the real-time PCR experiment. And then, as I mentioned, all of the links and the data that is shown in the pathway is supported by peer-reviewed publications, and they are shown at the bottom of the page, and they are hyperlinked to PubMed so that the researcher can easily find the primary data that supports these relationships. Next slide. So now looking at the Notch pathway in general, you can see jagged is at the top in the extracellular space, and then that activates Notch. And then Notch triggers a cascade that goes down to a transcription factor in the nucleus that causes transcription of HERP. And so HERP was the only gene that was interrogated in this pathway, and the color of this orange color indicates that it was upregulated in the experiment compared to cells that were grown in the absence of JAG. So this is consistent with the hypothesis, but again, interrogation of more of these genes may be interesting. Next slide. So now I'd like to go to the wheel chapter. And the wheel chapter is an excellent place to browse the biology that is trying to emerge from the data set that you've uploaded. So in the wheel, each circle represents a gene, and the colors of the circles represent the expression levels. In this case, yellow or orange is upregulated and blue is downregulated. And we also have the genes organized into these gray circles by molecular function. So molecular function could be kinase or transmembrane receptor, for example. And then, as you can see, we've selected one of the genes with black circle, and on the left there is a description of TGF-beta, which is the gene that's selected. Next slide. Around the rim of the wheel are interactive bands. If the user moves the mouse over these bands, then they can begin to browse their data directly from this interface. So within the purple processes section, there is a hierarchy that drills down to cellular migration. And in particular, there's cell movement of phagocytes, leukocyte migration. In particular, there's migration of endothelial cells, which seems relevant to this data set. Next slide. What we can do is we can click there and we can stay on the wheel view. And now we have fewer genes in our view. And again, we can drill down further. The genes that are shown are listed on the left, sorted by expression level, and it is very easy to click on any of these processes and pathways and diseases, interactions, and go to those relevant chapters to learn more. Next slide. If we select the development of blood vessels, which again is relevant to the vascular maturation that we're studying in this experiment, we can go to the process chapter. The process chapter shows all the genes that were interrogated in your experiment that are involved in development of blood vessels. And again, the publications are below. And in particular, if you go to the next slide, you'll see that integrin alpha V is one of the conclusions in this paper was that integrin alpha V was upregulated by Notch, and that was one of the key genes participating in the pathway that led to maturation of these cells. And it's easy in the process section to click on that gene and immediately see publications that are relevant to how integrin alpha V is involved in development of blood vessels. Next slide. Similarly to the process chapter, you can look at diseases that may be relevant to this biological system. For example, vascular disease. You can see on the left that there are 26 genes that are involved in this disease state, and the genes are colored by whether or not they have biomarker information, mutation information, drug target information, and by expression level as well. And then at the bottom of the page, you can see that there are various references for clinical trials that have been published investigating one or more of these genes as targets, and biomarker and mutation studies as well. Next slide. Often in gene expression experiments, we're looking at particular networks of genes that we suspect may interact with each other, of course. And so we provide an interface for viewing those interactions in the interactions chapter. Here I've chosen VEGF-A, and not only in selecting VEGF-A can you see all the interactions, both downstream in blue and upstream in red, between VEGF-A and the other genes in the data set, but also by hovering over and clicking on one of the links between VEGF-A and one of the other genes, you can see the publications that directly support that relationship. Next slide. And finally, the gene table. The gene table is a simple output that can be used, uploaded into a spreadsheet program like Excel so that you can continue to understand and manipulate the data yourself and document the data yourself. All of the figures and the tables in the iReport can be exported, as well as the publication lists. And if you look on the lower right of this screen, there's a section called Notes, and this section is available on all of the chapters. And as you discover a biological finding that's relevant to you or potentially begin to develop new hypotheses, You can make a note in this section, and then as you navigate away from that view and maybe return to looking at all the genes and go down a different path, that notes section will persist, and that can be exported as well. And when you come back to Ingenuity and log back in and view your report again, then those notes will persist. As I mentioned, we have focused on the ability to share these reports as well. And so there's a button not shown here to share these reports. If you share the report, the person who receives the report will also be able to see the notes that you've generated. So it's an excellent way to summarize your data for your colleagues, to quickly identify the key biological findings in your data, and have access to publications and references to help you prepare your own publication, your own poster and presentation. Next slide. So just in conclusion, I'm happy to introduce you today to Ingenuity iReport. It is fast, it's accurate, it's interactive, and it's customized. The way the product is constructed is that with any RNA-Seq data or microarray data, and we have file formats that are supported listed on our website, you can generate a free preview of your report. You can upload your data and see what the report will look like. And to do that, you can go to www.ingenuity.com/get-iReport. Real-time PCR is being commercialized in a partnership with Biotechnology. And so the URL to learn more about real-time PCR is www.lifetechnologies.com/iReport. And real-time PCR is currently in the beta stage of development and is being offered through early access for free. So I encourage you to go to this link and upload your real-time PCR data and obtain a free report that summarizes the biology behind your gene expression results. Next slide. Thank you all for your participation in this webinar. Thank you to the other speakers as well. And back to you, Tamlin.
And thank you, Gordon. Before we begin the Q&A session, I want to remind everyone that we still have time for more questions. So type your question into the Ask a Question box and hit submit. All right, so we have a lot of questions. Michael, we have a lot of questions for you specifically, so let's get started. What is the advantage of analyzing single cells when the objective is to mine genes?
Can you hear me?
Yep.
Very good. The advantage of analyzing single cells is that you reduce the complexity. When you treat any species or sample or cell culture, you will be changing gene expression globally, but different changes actually occur in different genes. So by looking at the genes in the different cells, you can separate genes that are involved in the different pathways.
Okay. How many cells do you need to analyze to get consistent data?
Yeah, that's a good question, and it's a difficult one. It depends on how different the cells are, but typically you need to analyze a minimum of about 50, 60 cells, and if they are like very similar and you want to see details, then you have to go up to 100 cells.
Okay. And how many genes can you study per cell?
We study regularly 25 to 50 genes per cell, but you can actually go up to 96, which is pretty handy in some of these high-throughput instruments.
Okay. How do you normalize the data?
That's an interesting question, but it's actually trivial because typically you normalize the data per cell. That's the most intuitive way to normalize the data.
All right, here's another question. On what basis was PCA chosen as a selection criterion for looking at different populations of cells?
That's very interesting, a very good question, and we are still learning. But typically the way I do it is that I inspect the loadings. That is that that's the weight factors. How important are the genes behind the PC vectors? And if you see that you have a separation, let's say along the PC1 axis, and you look at the loadings for the first principal component, and that will identify the genes.
Okay, Michael, what software are you using to conduct the pathway analysis and identify disease association of genes?
Well, I was certainly using the Ingenuity Report.
All right. I have a multi-part question for you now. Is it qPCR that you have to select the genes before starting the experiment? So essentially, you already know what genes are involved in which pathways before iReport, and what does iReport help exactly with when compared to IPA?
Well, I can take the first part, the iReport versus IPA. I passed Gordon. But I think it's a very, very interesting approach to use the iReport for qPCR data because we're using a different approach. When you work with microarray data, you typically do a whole transcriptome profiling. And then you are suffering from the challenge that there will be a large number of false positive leads there. When you work with qPCR, you work in the opposite way. You start with a very small number of genes, and which means that you don't have the problem of false positives. You establish that these genes are really relevant for your case. You inquire the iReport, and you find additional genes that may be relevant because of the correlation with those that you have already indicated. And then you include those in your studies. So essentially, you're working your way up, increasing the number of genes, in contrast to microarray studies where you actually start with a large number of genes and you're decreasing the number.
Gordon, do you want to weigh in on that question as well?
Sure. I can address the second piece around IPA and iReport. iReport, many of you are familiar with Ingenuity's product IPA. iReport is a different product from IPA. So iReport is not IPA, but it's a new product from Ingenuity. And iReport is customized to a specific experimental data set. There are several other differences as well. But I would just echo Michael's comment that with qPCR, start with a smaller number of genes, and then it can be important to understand the context of your results, the presence of genes that you actually did not interrogate, and other biological entities that you did not interrogate, cytokines. So our intent with iReport for qPCR is somewhat different from iReport for microarray and RNA, trying to biology.
All right. Gordon, was that it? Did I? Okay, Michael, I just have one more question for you then. Instead of correlation to identify genes, have you taken into consideration feature selection techniques, including machine learning?
We are doing that when certainly today when we analyze classical samples, for example, for disease classification, certainly. For single-cell profiling, it's not really clear how those more powerful tools can be used because as when you work with single cells, it's typically exploratory. You don't know really what you're looking for. So trying to detect and identify new features. But I'm sure we'll find ways how to use machine learning tools also in this aspect. Machine learning is, for example, available in GENNews. You can use that as a tool to select your genes and then to inquire the iReport, for example. Absolutely.
Okay, Milan, we have some questions for you now.
Sure.
The first one, first I compliment you, very nice data, but I did not hear a clear answer for why one should favor RNA-Seq over microarray if the appropriate cell types are compared. Can you address this?
Yeah, no, that's a great question. I actually get that quite often. So if you've got the appropriate cell types, then the advantages of RNA-Seq over microarray are the known technical and also biological advantages that you have with the platform. So with technical, you have less background. You have increased sensitivity, increased accuracy when you compare to gold standard TACMAN assays. In terms of the biological advantages, a lot of this has to do with the fact that I couldn't show all the data with only about 18 minutes here, but obviously increased ability to detect, to analyze alternative splice isoforms. Also, when we're looking at non-coding RNAs, we have a lot of analysis in long energetic non-coding RNAs that I did not present here, ability to pick up precursor microRNAs, and also if you're doing microRNA sequencing, small RNA sequencing, you can also analyze that. In addition, also RNA editing and several others. So I think when you're designing your experiment, if you feel that you would like those added advantages, that's when RNA-Seq will be superior.
Okay, Milan, why was TFBOXP3 upregulated in your triple-negative cancer cells compared to control, and does this result represent some cell contamination picked up by RNA-Seq data, or do you think this result is significant?
Yeah, very intuitive for the questioner. That's actually a great question. So I was actually very surprised too when I saw FOXP3. So when we designed this experiment, we really focused on very, very high tumor cellularity in terms of our triple-negative samples, over 90%. So what I think we're seeing with the FOXP3, like with triple-negative, that we see with a lot of triple-negative, we're going to see some, even if it's a minority cell population, we're seeing some lymphocytic infiltration. I believe the FOXP3 are probably coming from those T cells that are mixed in with those tumors.
Okay, one final question for you, Milan. As transcriptional factor, how do you explain the higher expression but predict inhibited?
Yeah, no, that's a great point too. So that's what I was trying to allude to, that obviously a lot of these transcription factors could be mutated, especially in the context of P53. So you may have higher expression, and this actually applies across cancers. I mean, you could have a higher expression of transcription factor, but if it's mutated either heterozygously or homozygously or with an dominant negative fashion, obviously then we would not see as downstream effectors being either upregulated or downregulated as we expect, and so thus the algorithm is going to tell us that it's inhibited.
Thanks, Milan. Gordon, we have a bunch of questions for you now. First, I guess a clarification. The person asks, the iReport can be used with microarray data, right?
Yes, that's correct. We actually have 3 flavors of iReport. iReport can be used for RNA-Seq analysis, for microarray analysis, or real-time PCR analysis. And we actually have some slightly different pathways to learn about all of those products. So to learn about microarray and RNA-Seq, go to Ingenuity.com. And then, as I mentioned in my presentation, real-time PCR is being commercialized in collaboration with Biotechnology. So there's a URL, www.biotechnologies.com/iReport.
And Gordon, when is the iReport for real-time PCR going to be available?
It is available now, and it's available for free for a limited time. So again, go to that LifeTech URL, and you can begin to upload your real-time PCR data. And it's free during this beta period, and then after that we will commercialize the product.
Hey, Gordon, can you explain how iReport is different from other approaches currently used to analyze expression data?
Sure. That's a great question, and we get that quite a bit. So there are, of course, many tools available for statistical analysis, so going from raw data to fold change, for example. In real-time PCR, this would be going from CT values to fold change or to RQ. There are also tools available to understand the pathway analysis of sets of genes after you've done that statistical analysis. So iReport is taking a new approach where we're trying to go all the way from the instrument to the biology. And that makes it different for a couple of reasons. One of them is, of course, it takes the whole process into account, but it's also really aimed at the bench scientists, the scientists that have the raw data, that are looking for the biology, and they don't necessarily want to dive into become experts in bioinformatics. And so it's meant to be an easy-to-use tool with a quick statistical analysis up front and rich biology at the end.
All right, Gordon, you said that outliers are removed. What if they belong to a really significant gene probe expression?
Great question. You know, in general, our philosophy in designing iReport was that we did not want to tell researchers how to analyze their own data, but we just wanted to provide an interface for them to analyze their data. And so we have some options for excluding outliers or not excluding outliers, and the severity with which outliers will be identified. And so you know your experiment best, so you can use that the outlier settings.
Okay, what are the optimal endogenous controls for the iReport output?
Yeah, that's another great question. So again, endogenous controls is a huge challenge in real-time PCR. We do not recommend specific endogenous controls. What we do is we provide an interface for you to see how the endogenous controls, any candidates you identify in your data set, how they vary across the various samples. And then we can also provide some scoring based on different methods for suggesting which endogenous to use, but the choice is up to the researcher.
Okay, and what is the file type I can upload for iReport?
So for real-time PCR in particular, data from any real-time PCR instrument can be used to produce an iReport. The iReport, however, is optimized for Applied Biosystems Biotechnology instruments. So if it's in those formats from AB or from Life Technologies, then you can upload the CT level data directly, or you can upload a fold change or RQ list. If your data is not in that format, if you're using it or if you prefer to analyze on the then we have templates where you can copy and paste your data into the templates and generate an iReport.
All right, Gordon, I have one more question for you. Can we insert log2 fold change data from RNA-Seq into iReport?
Good question. What I reviewed today was real-time PCR. So for RNA-Seq, it is a different pipeline for getting that data into the iReport. The iReport itself looks similar in many ways to our iReport that I showed you, but the pipeline is different. For RNA-Seq, we use CUPDIF file formats. And for more information, I suggest going to ingenuity.com/get-iReport.
Okay, thanks, Gordon. Milan, we just had one question come in. I thought we had a few more minutes. You could answer this. In light of recent breast cancer DNA sequencing studies, can you enumerate on how your expression studies in triple-negative breast cancer complement these recent results?
Yeah, that's a really great question. So I think probably many people on the line familiar with the fact that a lot of whole genome sequencing studies are being done in breast cancer through the TCGA consortium, also the ICGC. So when it comes to triple-negative breast cancer, the more recent results really show that out of somatic mutations, P53 seems to be the only really recurrently mutated gene triple-neg, at least across the board. Actually, a presentation at AACR last week it was a small tidbit, but it was some very exploratory analysis where it seems like there's an enrichment, some mutations in binding sites for retinoblastoma, and we saw in our case retinoblastoma being inhibited. So I think for what we know in terms of the mutational landscape of triple-negative currently with the genome sequencing studies, our expression data correlates well with what is known.
All right, Milan, thank you very much. And our time's up. Thank you for listening in. Thanks for the great questions as well. This webinar will be archived for 6 months on our website, www.GENNews.com. If you missed parts of it, you can watch it again, or you can recommend it to friends and colleagues. Once again, I want to thank our panelists as well as our sponsor, Ingenuity Systems. We'll also be sending you a survey shortly and hope you'll take the time to respond. Your comments help us continue to provide topical and timely webinars. Thank you.